how photosynthesis works? Or are you wondering how plants get their own food? For today's video, we will tackle this through this experiment.
Materials needed: transparent cups, permanent marker, baking soda, measuring cup, water, 1/8 or 1/4 teaspoon, dish soap, light source, puncher, plant leaves, syringe, aluminum foil, paper towels, and timer. First, label the 2 cups. The other one with baking soda and the other is without baking soda. After that, prepare the baking soda solution. First, add 300 ml of water. Then add 1/8 teaspoon of baking soda to the water and mix until the baking soda has dissolved. And after that, add 1 drop of dish soap to the baking soda solution and gently stir it until it has dissolved. After preparing the one with baking soda, let's now prepare the other. First, add 300 ml of water. And 1 drop of dish soap. With the help of puncher, cut 20 leaf teas from the plant leaves. The next step is to remove the plunger from the syringe and place 10 leaf teas. Place the plunger back into the syringe and push it down until only small volume of air is left. Suck up small volume of the baking soda solution into the syringe with the leaf teas. Carefully push all the air out of the syringe. Close the opening of the syringe with a finger and draw back on the plunger to create a vacuum. Hold the vacuum for 10 to 15 seconds and swirl the leaf teas to suspend them in the solution. Remove the plunger from the syringe and pour all the 10 leaf teas and the solution into the second cup. Fill the cup with baking soda solution up to the depth of about 3 centimeters. Cover the cup with aluminum foil so no light can pass through it. You want to keep your leaf teas in the dark until your another cup is ready. The next step is to remove the plunger from the syringe and place 10 leaf teas. Place the plunger back into the syringe and push it down until only a small volume of air is left. Suck up a small volume of the solution without the baking soda into the syringe with the leaf teas. Carefully push all the air out of the syringe. Close the opening of the syringe with a finger and draw back on the plunger to create a vacuum. Hold the vacuum for 10 to 15 seconds and swirl the leaf teas to suspend them in the solution. Remove the plunger from the syringe and pour all the 10 leaf teas and the solution into the second cup. Fill the cup with the solution without the baking soda up to the depth of about 3 centimeters. Remove the aluminum foil from the one with baking soda and place the both cups under your light source. Make sure that the light shines straight onto the cups from above. Start a timer. At the end of each minute, record the number of floating leaf teas from the both cups. Continue the experiment until all the leaf teas are floating in one of the cups. While waiting for the leaf disc, let's answer some questions. What do you notice about the leaf teas in the syringe? As I observe, when the leaf teas inside the syringe is combined with the solutions, it floats. However, when you put on the plunger, the leaf teas seemingly sat at the bottom of the solution. Don't forget to stir up the leaf teas so it won't stay at the bottom. How are your results different in the cups with and without baking soda? In the experiment I conducted, the two cups shows different results. The leaf teas in the solution with baking soda floats faster than the other without baking soda. In connection to that, as I conducted this experiment, the leaf teas in the baking soda floats. However, the other without the baking soda never floats. What does the vacuum do? As we all know, leaves naturally have small pockets of air between the spaces of cells, which is responsible to collect CO2 from the environment and is used in photosynthesis. In terms of that, when the leaf teas inside the syringe is sucked To create a vacuum, the air inside the leaf is forced out, causing the leaves to sink. What happens to the leaf disc? After the leaf discs undergo to the vacuum, it sinks at the bottom of the solution. Why do you think the leaf disc starts floating? During the leaf disc experimentation, the leaves are at the bottom of the solution. However, as the light goes through it, it starts to float. Why? It is because the air packets of the leaves are filled with baking soda, which stands as the carbon dioxide. As it goes through the process, it produces oxygen, which is the reason why leaf disc floats. When do 50% of your leaf disc float? After 20 minutes of waiting, 50% of the leaf disc in the solution with baking soda floats. In that case, the 100% of the leaf disc floats at the time rate of 36 minutes in total. As you may wonder, the whole process takes a lot more time than the others, which we can conclude that other aspects such as the light, amount of water, baking soda, and liquid soap is related to it.